All right, this is lecture 2 for chapter 1, and this is scientific method. So the first question about the scientific method is, why do we even have a method at all for our science, and do we need one? The answer is, well, the reason we have a method is it's a way of organizing our interest, our research, into such a manner that we can control our observations to give us data that is non-biased, give us information and conclusions that we can pass on to other individuals. It just allows our method to be a little cleaner and maybe more valid to have a clear method as to how we reached our conclusions. We're not guessing. We are trying to prove or disprove something whenever we start to do some sort of scientific inquiry. It must be scientific, it must be provable, it must be testable, which is sometimes actually really hard to do. You've all talked about scientific method before in other science classes, and it usually involves something like asking a question and then creating a hypothesis. There's lots of different ways we can do this. I'm going to go with 5 different steps in this, which is problem, hypothesis, experiment, data, and conclusion. 5, right? Sometimes you'll see other people say there's 6 or there's 4. It doesn't really matter. It's all pretty much doing about the same thing. So I'm not going to necessarily be too perturbed if you want to think that there's other different stages in here. But this is what we're going to roll with. So first part of this is problem. This is often stated as asking a question, like you have a problem that you want to solve, or there is a question you want to answer. That might be a very, very specific question, or it might be a general question. All right? That question has resulted from some observations you've made. You were observing something, you were taking qualitative and quantitative observations and getting some data about something. You said, why is this like this? Well, all of a sudden, there's your problem. That's what you want to answer. Why does that behave that particular way? Why is it like that? That's what stimulates the initial scientific method. I want to point out here, the scientific method is not really that different from any other problem solving that you would go through in the normal course of a day. You walk in a room, you flip on the light switch, the light doesn't come on, you say, I've got a problem, light's not on. You've already instantaneously kind of gone through a lot of the scientific method just when that light didn't come on, because you thought to yourself, you said, oh, the light didn't come on, the bulb's probably burnt out, I'm going to go change the bulb, and you do, and then you flip it, it comes on. You just went through the whole scientific method right there. So this doesn't have to be overly complicated. It really kind of makes sense when you lay it out. So you have some sort of problem or question, and then once you ask that question, you typically come up with some sort of hypothesis that would be your educated guess, is the term that we often use, as to why something's like the way it is. It's not a wild guess. You wouldn't wildly guess that, oh, my lamp didn't come on because the power plant blew up. That doesn't make sense. It's much more likely, based upon your experience, that simply the light bulb has quit working. So that is an educated guess. So that hypothesis is an answer to your question, a potential answer to your question. Sometimes you can have what's called a null, N-U-L-L, hypothesis, that is a way of looking at it where you must disprove, like, my null hypothesis is that the light bulb is not burnt out, whereas your hypothesis is that it is burnt out. So by disproving, if you could disprove that it is not burnt out, that means, right, that it is. And it's just a way of thinking about it. So if you hear me say or read something about a null hypothesis, it's like make your regular hypothesis and make the exact opposite hypothesis. That's your null hypothesis. Sometimes it's good to prove both, just to make your scientific method a little more thorough. Okay, so you make a hypothesis. Hypothesis must be testable. You can't make a hypothesis that you can't test, because then there's no way to prove or disprove your hypothesis. And a hypothesis is not just a statement of fact. Light bulbs are made out of glass. That's not a hypothesis, that's a fact. And that has nothing to do with answering your question. Now, a better hypothesis could be, my light does not come on because the glass on the light bulb is broken, right? So that is a statement of something you can prove or disprove, right? When you're doing science and you have a hypothesis, it should answer a new or novel question. Once again, restating known facts is not a hypothesis. And it must be testable. If you can't test it, it's not a good hypothesis. And surprisingly, it's actually surprisingly hard at times when you're doing scientific inquiry to come up with a really good hypothesis. Sometimes it's hard to make something that's testable. It's sometimes a challenge, and we'll practice that. Once you do have your hypothesis, you get to test it. That's what we would call your experiment. The experiment is totally driven by proving or disproving that your hypothesis is correct or not. And a lot of times it's wrong, and that's okay, right? Whenever you're doing an experiment and using the scientific method, you must use what we call a controlled experiment. That is, you only change one thing at a time. That way you can tell if what changes you made or what test you were carrying out actually had the effect on the thing you were testing. For example, back to our light. I flip on my light. Oh, it doesn't work. Problem. Hypothesis, I think the light bulb's burnt out. But then when you go to test that, you say, well, while I'm changing this bulb, I'll put in a new one of those flippy switch things that always breaks, and maybe we should put in a new cord, and we'll reset the breaker and the circuit board in the breaker box, and then you go back, turn on the light, and it works. Well, do we know which of those things that you changed actually had the result of causing it to work? We cannot say with 100% certainty which one of those factors, those variables that you changed, had the end result. So that would not be a good controlled experiment. In the scientific method, you would change one variable or factor at a time in order to determine which of those particular factors had an effect. Okay? So that is an example of a scientific experiment where you control all of those factors except for changing one factor at a time, and then you see what effect that has. Do not confuse the idea of a controlled experiment with, in an experiment, the controlled what we call a control, or the controlled test group, or the control group. A control in an experiment is that group you do nothing to be able to compare your test groups to. So an example of this is, I'm testing my new fertilizer. I made it myself, right? I'm going to test this fertilizer on some plants and see how it makes those plants grow. I'm not going to put it on all my plants. I'm only going to put it on, let's say, half of my plants. And those plants are going to be the test group. I'm testing my fertilizer to see if that one thing that I've changed makes it grow different. But I need something to compare that difference to. So I'll have my other half of my plants be my control group, where I will not do anything to. I will not change no experimental variables on that. That way, if this group does grow taller, uniformly taller than my control group, I can say with certainty that, hey, this fertilizer does make it grow. I'll keep all my other variables the same because it's a controlled experiment. They all get the same amount of water, same amount of light, same soil. Everything's the same except for that one thing I changed. My control group does not have that change in it. That way I can say, this absolutely is proven to make this growth happen. Or maybe my fertilizer totally kills all my plants, right? These are still fine, but these are dead. Okay, so my fertilizer's not a good thing. What if they both die? Well, I don't know. That doesn't prove whether my fertilizer is good or not, but it does prove that all my plants were not good test subjects and something was wrong somewhere in there. That's why we always want to have that control group in our experiment. Okay, whenever you're changing things, some terms that we look at, your independent variable is the thing that you are changing. Okay, so I added the fertilizer. That's the independent variable. Sometimes that's called the manipulated variable. That's what I change, I manipulate. All right, I add the fertilizer. That's what I'm changing. Okay. The dependent variable is what happens as a result of us changing the independent variable. So it depends on what we do, and then that's the result. Sometimes it's called the resulting variable. Because I added my fertilizer, they all died. Because I added my fertilizer, they all grew better than the control group. Right? That was what happened, growth or death in this case, was totally a result of what I did. So I did this to it, and then you had this resulting dependent phenomena occur. We experienced better growth. That increase in growth was the variable that resulted as a result of us doing our independent manipulation of something, right? So independent, you change. Dependent variable, what happens because you changed it. And we'll practice that as well. Right? Remember, you have to have a control group. Otherwise, whenever you do your independent variable, you really won't know exactly what result your independent variable had unless you can compare back to some sort of group that has no manipulation, no independent variable, that you can prove that what you did actually made a difference. Whenever you get data back from an experiment, you can get lots and lots of data, lots of information. Graphing is a great way of representing that data, graph. And it's a great way of easily showing that there is some sort of change or relationship between certain factors. You must have accurate data. If you inaccurately record data, your results are going to be all wrong, and all that experimenting you did is not going to really be very beneficial. Whenever you reach the end of the scientific method, you're going to reach a conclusion. So you've said, this is my problem. Here's my hypothesis, what I think the answer to this question is. You have tested it. Is my fertilizer is great for plants, is my hypothesis. It will make them grow faster or taller. And then you test that by doing your experiment with your variables and your control. And as a result, you get some data, and you look at your data, and your data tells you something, and that allows you to reach a conclusion where you say, well, yes, that does or does not support that hypothesis, is or is not correct, and that tells you something. Now, it's great if your hypothesis is supported. Like, does my fertilizer really make plants grow better? Yes, my data does support that. Now, what if it doesn't support that? Is that okay? Well, that is okay, because that allows me to form a new hypothesis. Like, my fertilizer is great herbicide. It kills plants wonderfully, right? Can I test that? Sure I can. Hey, I'm right, okay? You can change your hypothesis. You can change how you test your hypothesis. Having a failed conclusion or hypothesis that doesn't work out, or a conclusion that's wrong, or if you want to say wrong, that's okay, because that points you to areas of more investigation. And that's really what science is about evaluating all those possibilities.